RNA are made up of long chains of ribonucleotides, whereas DNA is made up of long chains of deoxyribonucleotides. Each DNA molecule has a phosphate group and they are made up of nitrogenous bases. So this is the pentose sugar. So in RNA, every nucleotide residue, it has a hydroxyl group which is present at the 2' position in the ribose sugar. Hello everyone, a warm welcome to today's session on chapter 6 of 2nd PUC, that is molecular basis of inheritance. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous chapter, that is principles of inheritance and variation, we had studied that Mendel had done genetic studies keeping garden pea plant as an experimental material, right? Wherein he said that there is something called as factors which are carried from the parents to the progeny, and those factors to some extent they get expressed in the progenies. But he did not know that those were called as genes. But later on, when scientists found out about DNA and all that, they understood about the genetic sequences and all that. So in this chapter, we shall entirely study about the DNA, the structure of the DNA, what are the sequences of the DNA, how it is arranged, and many more. So to begin with today's session, let us start about studying the nucleic acids, because the DNA is nothing but made up of nucleic acids. So to understand, first we need to understand where is the DNA exactly present. So a body is made up of tissues, right? And what are those tissues? Tissues are nothing but the group of cells that they are made up of. And what is there inside the cell? The cell contains a lot of cell organelles such as the nucleus, the mitochondria, the Golgi apparatus and various others, right? In that, nucleus is the one that is very important because that controls all the activities of the other organelles. So therefore, what is there inside the nucleus? Inside the nucleus, it is made up of chromosomes. What is there in the chromosome? In the chromosome, DNA is there. What is the DNA made up of? The DNA is nothing but made up of the genes. And what are the genes? They are nothing but the nucleotide bases or the nucleic acids. So therefore, nucleic acids are polymers of nucleotides. Polymers meaning they are long chains of nucleotides. So deoxyribonucleic acid and the ribonucleic acid, that is DNA and RNA, are the two types of nucleic acids that are found in the living systems because they are made up of nitrogenous bases. So what are these? We shall study about each of these one by one, wherein we shall concentrate more on the DNA because that is the genetic material that is present in humans. RNA, we find it in bacteria, viruses and all that, but in humans it is DNA that is the most important. So talking about the nucleic acids, I told you nucleic acids are nothing but they are made up of polymers of nucleotides, right? So they are of two types, that is deoxyribonucleic acid and ribonucleic acid. So talking about DNA, that is deoxyribonucleic acid. So DNA acts as the genetic material in most of the organisms. But what about RNA? RNA, that is ribonucleic acid, though RNA also acts as a genetic material in some viruses, mostly it functions as a messenger. In our body also RNA is there, that is the messenger RNA or the mRNA, right? mRNA gets formed during the process called as transcription, translation and all that, which you will study in this particular chapter itself. In the coming sessions we shall study about it. So at that time, if the codes that are present on the DNA has to be read, then RNA has to be produced. That mRNA is the one that will actually synthesize the proteins later on, and those proteins are the one which are utilized for various biochemical activities that are taking place in a body. So therefore, usually RNA in other organisms, they act like a messenger RNA, carrying the messages from the DNA to the respective parts or the cells. So RNA has an additional role as well. So therefore RNA, it functions as an adapter, it functions as a structural, and in some cases it functions as a catalytic molecule that speeds up the rate of a function of something else. So this is about the RNA. So therefore DNA is the basic genetic material that is present in most of the organism. RNA is usually found in viruses, but apart from that, RNA usually acts in other organisms. It acts as a messenger RNA. So talking about the DNA in detail. So this whole chapter we shall discuss about the DNA, its structure and all that. Again, DNA is also made up of polymers, that is long chains of deoxyribonucleotides. That is the difference between DNA and RNA. RNA are made up of long chains of ribonucleotides, whereas DNA is made up of long chains of deoxyribonucleotides. And the length of the DNA is usually defined or measured in terms of number of nucleotides. So how many nucleotides are there? There are different nucleotides that are present in DNA. So we have adenine, that is the nitrogenous bases that we call adenine, then thymine, then cytosine and guanine. So how many nucleotides they are made up of, or how many nucleic acids they are made up of? Based on that, counting that, we can find the length of the DNA. So, or based on a pair of nucleotide is referred to as base pairs. So, how many pairs of nucleotides are present on that? Based on that also the length of the DNA is calculated. So, therefore, this is also a characteristic of an organism, that is how much length of DNA is present in an organism, because all organisms will not have the same length of DNA. You might wonder that Drosophila is very tiny, but Drosophila has larger, the length of the DNA in Drosophila is larger when compared to some other larger organism. Humans, we look very tall like this, but the length of a DNA is not that long when compared to that of the length of the DNA present in a plant, that is Arabidopsis thaliana. So that is why the length of the DNA is also characteristic of an organism. So say for example, let's consider a bacteriophage. So what are bacteriophages? Bacteriophages are nothing but those viruses that attack the bacteria. They are the bacteriophages. So in these bacteriophages, say for example, the 5 into 174. The name of the bacteriophage is 5 into 174. This particular bacteriophage, so this symbol is nothing but 5. So the name of it is 5 into 174. So this particular bacteriophage has a nucleotide, that is the length of the DNA is 5000. It has a nucleotide. How many nucleotides are there in that? 5386. How did they find out how many that 5386 nucleotides are there? They counted how many adenine, thymine, cytosine, guanine it is made up of. Therefore, putting everything together, they found out how many are there? 5386 nucleotides it is made up of. And say for example, there is one more bacteriophage, that is bacteriophage lambda. It has same, both are also bacteriophages itself, but they are different. And that is why length of the DNA is also characteristic of an organism, because both are bacteriophages, that is same species, but that is same organism, but the species are different or their kind is different, wherein 5 into 174 bacteriophage has a nucleotide of just 5386, whereas a particular bacteriophage called as bacteriophage lambda, it has a nucleotide of 48,502 nucleotides or base pairs. And say for example, Escherichia coli. So what is this Escherichia coli? It is also a bacteria, E. coli, which is present in the intestine, that has a base pair of 4.6 into 10 to the power 6. So 4.6 into 6 times of it, that is 10 to the power 6 times base pair it has. And haploid content of human DNA. So human DNA, it is 3.3 into 10 to the power 9 base pair. So therefore, what did we understand from this? It is not the size of the organism that actually decides the length of the DNA, but it is the number of nucleotides that are present in the DNA, or the number of nucleotides that particular DNA strand is made up of, that will determine the length of the DNA. So, therefore, the length of the DNA is also a characteristic feature of an organism. So now let's study about the structure of the polynucleotide chain. So we know that the DNA is made up of chains of nucleotide, right? So what all it consists of, we shall look into. So nucleotide has nothing but it has three components. So what are the three components it is made up of? It is made up of a nitrogenous base. So the nitrogenous base that are present in DNA are adenine, guanine, thymine and cytosine. And it is made up of a pentose sugar. So, a pentose sugar means it has 5 carbon atoms in it. So, it is made up of a pentose sugar, and that is, I am talking about DNA. So, it is made up of a pentose sugar, but if it is a RNA, it is made up of a ribose sugar, and if it is a DNA, it is made up of a deoxyribose sugar. So, both RNA and DNA are also made up of pentose sugar, that is a 5 carbon compound, but if it is a RNA, it is made up of a type of pentose sugar called as a ribose sugar, and if it is a DNA, it is made up of a kind of pentose sugar called as the deoxyribose sugar. So, therefore, what type of pentose sugar is DNA made up of? It is made up of a deoxyribose pentose sugar. That is why DNA, it is called as DNA, deoxyribose nucleic acid, and it is made up of a phosphate group. So, this phosphate group is the one that actually forms the backbone of the DNA. So, there are two types of nitrogenous bases present in DNA, that is, they are divided as purines and pyrimidines. So, purines means it is nothing but adenine, that is A, and one more is guanine, that is G, and it is made up of pyrimidines. So, pyrimidines are also two. That is, if it is DNA, it is made up of cytosine and thymine, but if it is RNA, it is made up of uracil. Always remember, RNA will not have thymine. Thymine will be replaced by uracil, whereas DNA will not have uracil, it will have thymine. So, what are the 4 nitrogenous bases DNA is made up of? DNA is made up of 4 nitrogenous bases, adenine, guanine, cytosine and thymine. So, this is DNA. And what is the nitrogenous bases present in RNA? It is adenine, guanine, cytosine and uracil. There is no thymine in the case of RNA. So, therefore, what are the purines present in DNA? It is adenine and guanine are the purines. Cytosine and thymine are the pyrimidines. In the case of RNA, it is adenine and guanine are the purines, cytosine and uracil are the pyrimidines. So, that is how it is. So, cytosine is common. So, I've understood. Cytosine is common both in DNA and RNA, right? So, cytosine, so adenine and guanine is common in both in DNA and RNA. Likewise, the pyrimidine base that is there, that is cytosine, it is common in both DNA and RNA. But, so cytosine is common in both DNA and RNA, but thymine is present only in DNA, right? Thymine, the pyrimidine base, thymine is present only in DNA, and the pyrimidine base, uracil, is present only in the RNA and it replaces the thymine. So, that is the difference between the DNA and the RNA, or the nitrogenous bases of the DNA and the RNA, or in particular the pyrimidine base pair of the DNA and RNA. So, this is one of the important 1-mark question they will ask. So, which is the pyrimidine that is common in the case of DNA and RNA? It is cytosine. So, cytosine is the pyrimidine, which is the purines that are common, adenine and guanine, both in the case of DNA and RNA. So, therefore, what all we had understood? We had understood they are made up of a pentose sugar, which is a 5-carbon sugar, that is deoxyribose pentose sugar, and it is connected to a phosphate group. Each DNA molecule has a phosphate group and they are made up of nitrogenous bases. So, this is the pentose sugar. So, 1 carbon atom, 1, 2, 3, 4, 5. Therefore, it is a pentose sugar. So it is C5H2. So therefore it is a pentose sugar and it is connected to a nitrogenous base, that is adenine. Even here, pentose sugar, then thymine, then a phosphate group. Again here, a pentose sugar, then we have a guanine and a phosphate group. Again here, a deoxyribose pentose sugar, then a cytosine and a phosphate group. So the 5' and 3' and all that we shall study later when studying about the Watson and Crick model of DNA. So for now we need to understand what is a polynucleotide chain of DNA. It is a polynucleotide chain, right? Because it is made up of many nucleotides or nitrogenous bases. So it is made up of chains of nucleotides. So what are the chains of nucleotide consists of? The chains of nucleotide consists of a pentose sugar, it consists of a phosphate backbone, and it consists of the nitrogenous bases such as the adenine, thymine, guanine and cytosine. If it is a DNA. If it is a RNA, it is made up of a ribose sugar and it is made up of the nitrogenous bases, that is adenine, uracil, guanine and cytosine. So that is the difference here. So this is about the structure of the polynucleotide chain. Next, moving further. Now a polynucleotide chain has to be formed. If these has to form chains, then they have to be connected to one another, right? So what is it we shall look into? So a nitrogenous base is linked to the pentose sugar through a N-glycosidic linkage to form a nucleoside. So what is a nitrogenous base? Nitrogenous base is just this adenine. Adenine is a nitrogenous base. Then here thymine is a nitrogenous base. Then here guanine is a nitrogenous base. Then here cytosine is a nitrogenous base. Now these nitrogenous bases as such, are they present in DNA? No, they have to be linked, right? To what are they linked? They are linked to the deoxyribose pentose sugar. How are they linked? They are linked to the pentose sugar by a bond. What is the bond? The bond is nothing but a glycosidic linkage. So they are linked by a glycosidic linkage, and such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. So that is what they are made up of. So now we have understood that the nitrogenous base is linked to this pentose sugar by what type of bond? It is linked by a N-glycosidic linkage bond, right? N-glycosidic bond. So everywhere thymine is linked by N-glycosidic bond to this particular pentose sugar, deoxyribose pentose sugar, like similarly guanine also, similarly cytosine also. Now, are only these two present in this particular nucleotide chain? No, a phosphate group is present, right? How is this phosphate group linked? The phosphate group is linked to the 5' OH of the nucleoside through a phosphoester linkage. So this phosphate group is linked to the 5' end. How is it linked? It is linked by a bond called as phosphoester. So phosphoester linkage or phosphoester bond, and also it is linked to the corresponding nucleotide. That's true, right? Phosphate is linked to the nucleotide. What is a nucleotide? Nucleotide is nothing but a nitrogenous base connected to a pentose sugar. That is deoxyribose pentose sugar. It is a nucleotide. Now, the phosphate is linked to the nucleotide as well as it is linked to the 5' end. It is linked to the 5' end by what type of bond? It is linked by a phosphoester bond, and it is linked to a nucleotide or it is linked to a deoxynucleotide depending on the type of sugar that is present, and therefore a particular linkage is formed. Now, this is about how they are linked. Now, how are the two nucleotides linked? So now we understood. So just be careful here about the different bonds that are there. So now we understood adenine is linked to the deoxy pentose sugar by a bond which is called as N-glycosidic bond. Now what is this? It has formed a nucleotide. Together it is called as a nucleotide, right? Now this whole nucleotide is linked, that is the phosphate end of the nucleotide is linked to the 5' end of the DNA by a bond which is called as phosphoester bond. Now one nucleotide is linked to the other nucleotide by a bond. What is that? That bond is called as, so two nucleotides are linked through 3' to 5' phosphodiester linkage. So they are linked by 3', 5' phosphodiester linkage, and more nucleotides can be joined by such manner. That is how one nucleotide is linked to the other one is linked to the other, therefore forming a polynucleotide chain. So you have understood, right? So we understood the linkage between the pentose sugar with that of the nitrogenous base and that of the phosphate group, that is N-glycosidic bond. Now this phosphate group is linked to the 5' end by a phosphoester linkage, and each of these nucleotides are interlinked to one another, and the bond depends on the type of sugar molecule they are made up of. So that is how each of these nucleotides, they link to one another, forming a polynucleotide chain. Therefore, DNA is a polynucleotide chain made up of nitrogenous bases, a deoxy, pentose sugar, and a phosphate group. So this is about the structure of the polynucleotide chain. So next, studying about the structure of the polynucleotide chain further. Therefore, now a polymer is formed. What is a polymer? Chains of nucleotides are attached. Therefore, a polymer thus formed. At one end, it will have a free phosphate moiety, that is at the 5' end of the ribose sugar, which is referred to as the 5' end of the polynucleotide chain. So therefore, we had understood the phosphate group is linked to the 5' end by a phosphoester bond, right? Now therefore, a polymer that is formed, this chain that is formed, on one end the phosphate group is free because it is not linked to any of the nucleotides. But what is it linked to? It is linked to the 5' end of the DNA strand. And similarly, at the other end of the polymer, also there is no free phosphate group, but here on the other end of the polymer, the ribose sugar is linked to the 3' end of the hydroxyl group, which is referred to as the 3' end of the polynucleotide chain. So always understand, in one end of the DNA, the phosphate group is free because further it is not linked to any of the nucleotides. But what is it linked to? It is linked to a 5' phosphate group, therefore forming the 5' end of the DNA. And on the other end of the DNA, the ribose sugar is not linked further, it is free. To what is it linked to? It is linked to the 3' hydroxyl group. So therefore forming the 3' end of the DNA. So these are the two ends of the DNA, and the backbone of a polynucleotide chain is due to the formation of sugars and the phosphate. So therefore, sugars and the phosphate, in this particular case, the deoxyribose sugar and the phosphate that is there, they form the backbone of the DNA, and it is this phosphate that actually decides the helical structure of the DNA, and the nitrogenous base linked to the sugar moiety project forms the backbone. So therefore, the backbone of the DNA is made up of the phosphate group and also the sugar molecule. So this was about the structure of the polynucleotide chain. Next, moving further, let us study about the structure of the polynucleotide chain in the case of RNA. So far we had studied the structure of the polynucleotide chain in the case of DNA. So in RNA, every nucleotide residue, it has a hydroxyl group which is present at the 2' position in the ribose sugar. And also in RNA, the uracil that is there, it is found in the place of thymine, and also so that is also one of the difference. So that just that difference, apart from that, rest all it is same. Only thing is an additional OH group will be present in the 2' position of the ribose sugar, and one more thing is the uracil that is found, uracil is usually found at the place of thymine. That is, instead of thymine here, there will be uracil. That is the only difference that is present. And say for example, we have 1 prime, 2 prime, 3 prime in the sugar. Here in this particular 2 prime end, one OH group will be present. Where? In the case of RNA. But in DNA, all this is not there. So that is the difference. So that is the only difference between the structure of the polynucleotide chain in the case of RNA and in the case of DNA. So next, moving further, let us study about the contributions of various scientists towards finding out the structure of the polynucleotide chain. So the DNA as an acidic substance, it is present in the nucleus. So who discovered that? Who said that DNA is an acidic substance that is present in the nucleus? Otherwise, people would have not come to know, right? So it was first identified by a scientist called as Friedrich Mischer in the year 1869. So this was the scientist, that is Friedrich Mischer. He was the first person to have identified it in the year 1869. And he was the first person to name it as nuclein. He didn't call it as nucleic acid or he didn't call it as the DNA, but he named it as nuclein. However, due to some of the technical limitations in isolating that the polynucleotide chain and all that, the proper structure of the DNA could not be put forward by further scientists. And later on in the year 1953, it was James Watson and Francis Crick. That is why it is called as Watson and Crick model of DNA, because they were the ones who actually put forward the structure of the DNA, because the microscopes had developed and all that. So James Watson and Francis Crick, they actually put forward the structure of the DNA. And based on the X-ray diffraction data that was obtained, that was X-ray diffraction data, it was found out by Maurice Wilkins and Rosalind Franklin. So these two, they actually developed this X-ray diffraction data, and using this X-ray diffraction data, Watson and Crick was able to find out the proper structure of the DNA. So therefore, the first identification of DNA, the credit goes to Friedrich Mischer, because he was the first person to say that something is present, an acidic substance is present inside the nucleus of the cell, and he called it as a nuclein. But he was not able to give a proper structure to it because various instruments or various advanced tools were not available during his time. So later on in 1953, it was few of the scientists, that is Maurice Wilkins and Rosalind Franklin, these two scientists, Maurice Wilkins and Rosalind Franklin. So these two scientists actually, they developed an instrument which actually was based on X-ray diffraction data, and using that X-ray diffraction data, James Watson and Francis Crick, they were the ones who found out the actual structure of the DNA and said that DNA is a double helical structure. So they actually, Watson and Crick proposed the double helix model of the DNA. So let's study about the double helix model. So one of the hallmarks of their proposition, that is Watson and Crick, was that the base pairing between the two strands of polynucleotide chain. That is true. Because of the base pairing only, the DNA is called like this. Say for example, we have adenine here, then thymine, then cytosine, guanine. Because of the base pairing occurring between these, that is why the helical structure of the DNA is formed. However, this proposition was also based on the observation of Erwin Chargaff, that he was the one who said that for a double-stranded DNA, the ratio between adenine and thymine and the ratio between guanine and cytosine are constant and equal. Means adenine always base pairs with thymine, cytosine always base pairs with guanine. It cannot be different. Adenine cannot base pair with cytosine, nor thymine cannot base pair with guanine, nor neither way, because it should always be complementary like this. So that credit goes to Erwin Chargaff. So the credit of discovery of the structure of the DNA, that it is a double helical structure and the base pairing occurs between the two polynucleotide strands, all that goes to Watson and Crick. Whereas the reason why it is double stranded and how exactly the base pairing will take place, all that goes to Erwin Chargaff. And the base pairing, it actually confers a very unique property to the polynucleotide chain. So, the DNA is double helix only because of the base pairing, and it is the base pairing is also one of the characteristic feature of the DNA. So, they are said that they are said to be complementary to each other, and therefore, if the sequence of bases of one strand is known, then the sequence of other strand can be predicted. That is true, right? Because say for example, I'll write a sequence here, A, T, G, C, T, T, G, A, C, C. Now, I can easily predict the sequence of the other strand. Why? Because adenine pairs with thymine and thymine pairs with adenine, guanine with cytosine, cytosine with guanine, then thymine here adenine, again adenine, then here cytosine, thymine, guanine, guanine. I could predict, right? So that is what they said. So knowing what type of nitrogenous base one strand is made up of, because of its complementary property, we are capable of predicting what the nitrogenous base might be present in the other strand. So that credit and all goes to Chargaff. So this was about the nucleotide. And also, if each strand from a DNA acts as a template for synthesis of a new strand, the two double strand thus proposed would be identical to the parent till DNA. So it's also true. Why? Because again here because of base pairing. So say for example, there is this strand of DNA. Now this strand, they will undergo a process called as replication, wherein they will get separated, and for this parental strands, new daughter strands will form like this. Now when these daughter strands are forming, they will also be coiled manner to the parent DNA strand because of the base pairing character. So that is why base pairing is one of the important characteristic feature of the DNA, and because of this the genetic implications of the structure of the DNA became very, very clear. So therefore, thanks to Watson and Crick and also one more scientist, Chargaff. So next, moving on, we shall study about the salient features of the double helical structure of the DNA. What are the important characteristics of the double helical structure of the DNA? So double helical, so double, therefore it is made up of two polynucleotide chains, where the backbone is usually made up of a sugar phosphate. That is true, right? It is made up of two polynucleotide chains, where here one, this is the first chain, this one is the second chain, and base pairing is also seen. Apart from that, the two polynucleotide chain, what is the backbone of the two polynucleotide chain? It is made up of a sugar phosphate, right? So this is the sugar, this is the phosphate group. So it is made up of the sugar phosphate, and the base, nitrogenous base, this one, it projects inside. That is true. So therefore, this forms the backbone, the nitrogenous base projects inside. And the two chains have anti-parallel polarity. It means if one chain is from 5' to 3' direction, the other chain is from 3' to 5' direction. So you can see here, the first chain is from 5' towards 3'. The second chain is from 3' towards 5'. So that is the difference. That is why they are coiled in nature. That is why they exhibit polarity. So this is about the strand. Moving further, the bases in the two strands are paired through hydrogen bond. Can you see here? These are the two bases that are present. What did I tell you? They are made up of sugar. The backbone is made up of sugar and a phosphate group, right? And inner to that, towards the inside of the DNA, they have the bases, nitrogenous bases. So here, adenine, what is complementary to adenine? It is thymine, right? This is also a base. Now they are held together. Can you see here? How are they held together? They are held together by hydrogen bonds. Hydrogen bonds actually holds them together. So adenine is held by a double hydrogen bond to thymine. Guanine is held by a triple hydrogen bond to cytosine. So that is how they are held. And adenine forms, that is what I said here, adenine forms two hydrogen bonds with thymine, and whereas Similarly, thymine forms 2 hydrogen bonds with adenine, and guanine is bonded with cytosine with a 3 hydrogen bond, and also cytosine is bonded with guanine with a 3 hydrogen bond. So, if you can see here, cytosine and guanine, they have 3 hydrogen bond. Adenine thymine or thymine adenine, they have 2 hydrogen bonds. So, that is how they are bonded, and this always a purine comes opposite to a pyramid. That is true, right? Because a purine is always opposite to a pyramid in here, and this generates approximately uniform distance between the 2 strands of the helix. So, this is the structure of the DNA helix. So, what is it made up of? It is made up, the backbone of the DNA is made up of a sugar and a phosphate group, and towards the inside of the DNA, the nitrogenous bases are present complementing to the 2 strands, wherein adenine is held to the thymine by a double hydrogen bond, and guanine is held to cytosine by a triple hydrogen bond. And always the DNA, they have a polarity because they have the strand from the 5' end to the 3' end, and one more strand is from the 3' end to the 5' end. So, this is about the salient features of the structure of the DNA. So, next, moving further, let us study about their coiling. It is a double helical structure, right? So, they need to coil. How do they coil? So, the 2 chains are coiled in a right-handed fashion, and the pitch of the helix is 3.4 nanometer. That is, the pitch is somewhere between 3.4 nanometer. So, and there are roughly 10 base pairs in each turn. So, in each turn, how many base pairs are there? So much are the base pairs. How many are there? 10 base pair. Here one turn is there, that is also 10 base pair. Here one turn is there, that is also 10 base pair. So, for every 10 base pair or every 10 nitrogenous bases, the DNA will coil. So, that is about it. Why do they coil? Again, because of the polarity. So, that is the reason. And what is the angle of coiling? It is about 3.4 nanometer. And consequently, the distance between a base pair in a helix is approximately equal to 0.3 nanometer. So, a distance in the base pair. So, the distance that is between one base pair and the other base pair is approximately 0.34 nanometer. Distance between the base pair. So, distance between, so say for example, the distance between adenine and thymine, or the distance between adenine and guanine, all that, it is somewhat between 0.34 nanometer. And the plane of one base pair stacks over the other in a double helical manner. That is 2, right? So, this one, so it is stacking about one another, that again, so therefore, they are stacked one above the other, as you can see here. This stacking one above the other is the one that actually results in the formation of a double helical structure, and the plane of one base pair stacks over the other in a double helix, and this is the addition to, and this along with the formation of the hydrogen bonds between the base pair, it actually gives stability to the helical structure of the DNA. So, this is about the double helix. And also, we understood they are made up of base pairs. The base pairs are held to the, or the nitrogenous bases are held to the pentose sugar. The pentose sugar has a phosphate group. That phosphate group, pentose sugar, forms the backbone of the DNA, and the 2 nitrogenous bases on the opposite strands are held together by hydrogen bonds. And apart from that, for every 10 base pairs, the coiling of the DNA will occur, and the DNA exhibits polarity because of one of the strand is from 5' to 3' end, and the other strand is from 3' to 5' end. And the distance between the nucleotides is about 0.34 nanometer, and each nucleotide is stacked the pair of nucleotides and the strands are stacked one above the other. That is why they form a helical, like double helical like structure. So, all that we have understood. So, this was about the structure of the DNA, and this structure of the DNA, the complete credit goes to Watson and Crick, because they were the ones who suggested that DNA is a double helical structure. So, it is helical in nature and it is made up of 2 strands. Next, talking about one more important concept that you need to know before moving on to the further sessions of this chapter. That is called a central dogma. Central dogma is one of the basic concepts in biotechnology, which every student, biology students needs to know about it. So Francis and Crick, they proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA and a protein. That is right? Why? Because we have the DNA strand. In this DNA strand, the RNA strand gets produced. The RNA strand will be complementary to the template strand of the DNA. This RNA strand will come out, and this RNA strand, based on the sequence it is made up of, it will start to produce the proteins. So this is what we shall learn in the coming sessions, wherein you will study under transcription, translation and all that. This central dogma principle is the one that is followed. What it means? It means that DNA will undergo a process called as replication, wherein after replication, after unwinding of the DNA, because DNA is held together, right? The two strands of DNA are held together. Those strands should get unwound by the breakage of the hydrogen bonds which are present between the nitrogenous bases. When they get unwound, they follow a process called as transcription. During the process of transcription, the mRNA strand will get produced. This mRNA strand should translate the message of the DNA into a protein. That is how the protein gets produced. So this occurs in three important steps: replication, transcription and translation. Therefore, at the end, formation of the protein. So this is nothing but the central dogma. And in some viruses, the flow of information is in reverse direction. That is, from the RNA, the information is to the DNA. Rest all of the organisms, the information is carried from the DNA to the mRNA. But in the case of viruses, the information is carried from the RNA to the DNA. So this is about the central dogma principle. So this was about this particular session today. In the coming session, we shall discuss about, in further, let's study about the packaging of the DNA helix. So DNA, they are very, very lengthy in our body. But imagine if it is so lengthy, will a human body be enough to fit a DNA in each cell? Not possible, right? So that is why the DNA sits packed inside the cell. How exactly they are packed inside the cell, we shall look into, and we shall also study how exactly the history of DNA, that is, how DNA came into existence, how people came to know about the DNA. So we shall study about the search for the genetic material, that is nothing but the DNA. So all this we shall study in the coming session. So I hope you understood the session well. So we shall meet again in the coming session. Thank you.